and want to say thank you to the students that nominated me to come, and Sarah for inviting me, and a lot of friends and former colleagues in the audience. So I thought I'd start by telling you a little bit about my scientific journey. So when I started as an assistant professor at the University of Arizona, my good colleagues and mentors, Paul Brooks and Tom Meixner, invited me to participate in our first critical zone observatory proposal. This was a cross-campus, big interdisciplinary project that was luckily funded by NSF for about 10 years. And we worked on the combination of water, rocks, and life in the relatively shallow subsurface. And I was always curious how deep the critical zone extended. And I loved that type of interdisciplinary work. And that led to the next big interdisciplinary project that I helped to get off the ground and has been funded for about the last 6 years by a Canadian nonprofit called CFAR. It used to be the Canadian Institute for Advanced Research. And our project is called Earth-4D, Subsurface Science and Exploration. And this is an equally interdisciplinary project that we're studying water, rocks, and life in the deep subsurface on Earth as an analog for what you might find on other planets. And that project really made me think much larger scale, more planetary scale. And that introduced me to a colleague, Maggie Osborne, who's a geomicrobiologist that I continue to work with. And I then, after that project that's still ongoing, I got started in the Colorado Plateau by being invited by a colleague, Pete Rainers in geosciences, who was working with Eugene Szymanski and others on fluid rock reactions in the Colorado Plateau, and specifically looking at how all of the bleaching of the red bed sandstones that Dr. Brynle Beitler-Bowen and Margie Chan had studied so nicely, how that set the stage for later fluids that brought in copper that precipitated mineral resources. So we started to look at the sequence of events. And that was a really fascinating project, had some great students graduate that I'll show you their results. But what was missing from this project was the role of biology. And so about 5 years ago now, I led a team of interdisciplinary scientists. We got funded by NSF through their Frontier Research in Earth Systems, or FREST program, on a dream project called SMERFs, and it stands for the Evolution of Subsurface Microbe Rock Fluid Systems. And so I'm going to talk to you today about what we've learned over the last 5+ years in the Colorado Plateau, knowing that many of you are far more experts, and I'd be curious to hear your feedback. So here's a picture of one of our field glamour photos of all the interdisciplinary scientists, really long list, but the co-PIs on the project are Pete Rainers, who's a geochronologist, thermal chronologist, Basil Tikoff, who is a structural geologist, Maggie Osborne, a microbiologist, Henrik Drake, who studies tiny things, minerals, to look at past history, and then Grant Ferguson, who is a physical hydrologist and was the Darcy lecturer who came to visit you, I guess, in the recent past. So our SMERFs project, we were really interested in how the Earth's crust, as we see it exposed at the surface today, has been deeply buried in the geological past, and what that did in terms of the temperature and fluids, and recently was brought back up to the surface through exhumation, uplift, erosion. How that movement of the Earth's crust up and down may have changed the subsurface hydrology and microbial activity. We also know that the Earth's crust has experienced a lot of extension and contraction that may have opened up permeable pathways for fluids to migrate, and possibly those fluids bringing microbes back into the subsurface. And then we know the hydrology of the Earth's surface has changed, as topography has gotten steeper or more subdued, as seawater has inundated the landscape, and as in the more recent past, how glacial periods have led to wetter and colder climate. So all of that has impacted drivers and transport of fluid, heat, and solutes through the subsurface, at times through permeable pathways like faults and fractures, and reacted with rocks and minerals that may have changed the environment for microbial communities at depth in the Earth's crust. There were 3 driving questions of our SMERFs project. The first one was, how did these geologic forcings, specifically burial and denudation, driven the deep biosphere? What hydraulic gradients and permeabilities and fluxes are required to maximize the potential for subsurface microbial life? And finally, what evidence do we have from the rock record of past fluid flow and past microbial activity? So all of those driving questions kind of fit at the time when the proposal was funded with some big overarching questions that the National Science Foundation had been looking at from the Vision for Earth Sciences report. It's getting a little old now, but some of the topics that came up in that visionary report were looking at the importance of geologic time on these processes, the connection between the Earth's surface and the deeper interior, and the coevolution of geology and life. And some specific questions were, what are the causes and consequences of topographic change? How do biogeochemical cycles evolve over geologic time? And how do geological processes influence the biodiversity of the subsurface? And finally, this fit with NSF's push in really growing convergence research, which is very close to my heart. I love working on interdisciplinary teams of scientists. I think that far more interesting science happens at those intersections, working with other really productive and curious and creative people. And also they're understanding the rules of life. So we should all care about the subsurface because we've now produced hydrocarbons for over 100 years that has emitted CO2 and methane, has led to global warming. We're now in the climate crisis, needing to put some of that carbon back into the subsurface. We also are needing to explore and produce more mineral resources from the subsurface to meet our growing energy demands and to also help with the green energy transition. Geothermal energy, which I know happens in the state of Utah, is increasingly important, not only to meet energy demands, for homeowners and industry, but even in Arizona for things like data centers for cooling. And finally, we're storing, we need to store carbon dioxide from the atmosphere in the subsurface. We also have not solved the problem of nuclear waste storage. Other countries like Finland and Sweden are putting it in the subsurface. And we also need to store hydrogen. So hydrogen that we produce at the surface, we need to store it in the short term in the subsurface, like in salt caverns. And there's also natural hydrogen that is generated in the crust that we could produce for energy. Our use as humans of the subsurface is only accelerating. So again, over the last 100 years, we've produced a lot of fluids, both oil and gas, as well as co-produced waters. We've actually re-injected more water than we've produced from the subsurface, and that's for enhanced oil recovery. And all that movement of fluids has changed local redox conditions for microbial communities. And if we go towards the future and we make projections about how much fluid is going to need to be injected and produced from the subsurface, the flow rates are going to dwarf the natural background movement of water in the subsurface. And I think a really interesting fact is if you project how much fluid would need to be produced to get lithium from brines, it dwarfs the amount of fluid that's produced with oil and gas over the last 100 years. So before we start mucking up the subsurface even more, I think we need to understand natural processes in the subsurface to try to understand the fate of things like storage of hydrogen and carbon dioxide. So that brings us to our studies in the Colorado Plateau. It's your background, it's your backyard, it's our playground. We had started working in a place called the Paradox Basin, which is a subset of the Colorado Plateau. It is a foreland sedimentary basin. It's outlined in yellow because it's defined by the extent of the Paradox Formation's salt. We started working there because there's so many manifestations of paleofluid flow that have done important and interesting things, like bleaching of the former red bed sandstones that Brenda mapped out, I think in your dissertation or later, shown in the purple. And that area of bleaching again set the stage for copper and uranium and vanadium deposits. We also see a lot of accumulation of carbon dioxide, of helium, as well as oil and gas. So lots of yummy fluids moving around, doing interesting things that we could study. As Sarah described, I'm a hydrogeochemist, so I like to sample water. I tend to like to sample things as deep as I can get, and I sample the chemistry of the water, the isotope composition, the age, and then I work with microbiologists who study the microbes living in the water. And I tend to study the things that they produce as a biosignature. So this is a cross-section simplified of the Paradox Basin, going from the high elevation in the Upcombadre uplift. You've got over 3 kilometers of sediment overlying the basement rocks. An important unit is the Pennsylvania Paradox salt. It's over 1 kilometer in thickness, and it started to move early on in the history of this basin, and it forms a lot of salt diapirs, which I'm told I should call them salt walls, that come close to the surface. And then there's these interesting volcanic laccoliths that come up like the La Salle Mountains that create internal topographic highs. So my students have sampled water from the surface of the Paradox Basin in rivers and springs to shallow groundwater, so fresh to brackish aquifers from monitoring wells to water production wells, to then deeper samples from oil and gas, so the water produced with that, all the way down to the bottom of the sedimentary pile in the basal aquifer. We also were lucky to be able to sample a lithium exploratory mining well that was producing brine from within the salt. There's interbedded shale layers. So that was that salt brine sample. So I'm going to take a step back and tell you a little bit about what we know about the history of the rocks in the Paradox Basin, and specifically the temperatures that they reached in the geological past. So this is a burial thermal curve from a specific location. This is the tar sand triangle. If we go back to the time of deposition, I just want you to follow the unit in yellow. That's the white rim sandstone. It contains the tar sands today. I want you to see that after it was deposited, it was relatively cool temperatures until this really important period when the Mancos Shale started to be deposited and you reached maximum burial. At that time, the right rim sandstone sediments reached a relatively high temperature. What's important to know is that microorganisms that degrade oil and gas live up to about 80 degrees C. Beyond that, they're really inhibited. The maximum temperature of life on our planet has been found up to about 122 degrees C. So at this time period of maximum burial, the white rim sandstone would have reached a temperature that would have been too hot for microorganisms to be very active. I called it sterilized. My geomicrobiology colleague says, don't say that. Microbes are invincible, but they definitely would have been really inhibited. And this burial thermal history is supported by the vitronites reflectance in the shales, but also fluid inclusion data gives you temperature and clumped isotopes of carbonates. Another really important time period was the last few million years when you had incision of the Colorado River and massive denudation. So it brought that sediment that had been sterilized back up to the surface at cool temperatures. Again, the white rim sandstone likely reached temperatures beyond the limits for microbes that could degrade hydrocarbons. But yet, today, we see biodegraded oil in that sandstone, and there are isotopic evidence that microbes degraded oil coupled to bacterial sulfate reduction from carbon and sulfur isotopes. There's evidence that microbes are doing things today, but would have been sterilized in the past. If we take a bigger view and we look at other oil and gas reservoirs in other places throughout the Paradox Basin, most of those oil and gas reservoirs also reached these really high temperatures. And so thus, if we see microbial communities today from fluids produced from these oil and gas reservoirs, our hypothesis was that they must have been reintroduced relatively recently and by meteoric influx. So that time period was not interesting for microbes. They were either dead or inhibited, but it was an important time period because when it got really hot, hydrocarbons were generated and they were expelled from the source rocks, likely the organic rich shales within the salt. And it moved those hydrocarbons into formations that were above and below the salt. And so it brought those hydrocarbons much closer to the surface. And as I said, maybe primed those sediments for later microbial activity. This was work done by Lydia Bailey, who was a PhD student on our SMERS project. She was studying a specific location that I'm sure that Brenda has studied called Rainbow Rocks. It's a beautiful exposure of bitumen, residual oil, that has bleached the red bed sandstone, the Entrada Formation. And you can actually see from space that bleaching from the migration of hydrocarbons up the Moab Fault and laterally out into the sandstone. It was also a time period that it was not only hot and hydrocarbons generated and migrated, but it was also a time period when really saline fluids that were deposited with the salt were retained at depth. Oh, and I should have mentioned that Lydia went on to do a postdoc at Harvard and then interestingly is working for a carbon sequestration startup company in Boston. So we can see that time of maximum burial and retention of really saline fluids based on the chemistry of the brine that's produced from within the Paradox Formation. So this is a brine coming from the King Creek Shale interbedded. I call it the mother brine because it is the most salty fluid I've sampled in the Colorado Plateau. It's over 10 times the salinity of seawater. And we can tell that it came from evaporated seawater because it's really rich in bromide. It has very low chloride to bromide ratios and low sodium to bromide. We can also tell from the stable water isotope values. So you not only had retention of evaporated Paleozoic seawater that was deposited with that salt, you also have that brine and formations that are above and below the Paradox salt. So again, that was a really important time period, that deep burial. And then all of a sudden, something really dramatic happened just in the last 10 million years of the Colorado Plateau. You brought that sediment back up close to the surface because of incision of the Colorado River. So as the Colorado River started cutting down through this 3 kilometers of sediment, it created a massive drain that accelerated subsurface groundwater circulation. And it also created this huge topographic gradient from the top of the mountains, like the La Salles, to the bottom of the Colorado River. And it also unroofed the sediment. So if we go back to the Paleocene, this would have been almost maximum burial, where you had the thick Manco shale and gray capping all of that deeper sediment. And that was that time where it was too hot and it was too saline for microbes. But with incision and denudation, you unroofed all of that sediment. You got rid of that shale confining unit. You brought deeper sediment close to the surface that's now exposed today. It became in contact with circulating shallow groundwater. And it also brought that salt close to the surface where we see it exposed today. And that circulating groundwater is dissolved salt. and I'll show you evidence of that. So we can see the dissolution of salt in the recent geologic past because there are brines in the Paradox Basin that are the opposite of Paleozoic evaporated seawater. These brines indicate halite dissolution, really high chloride and sodium, relatively low bromide. We also see their stable isotopes look like meteoric water. So where are those brines? Interestingly, we find these salt dissolution brines not only near the surface, as we would expect interacting with these salt diapirs, but we actually see them also at the very bottom of the Paradox Basin. That's important because when there's salt dissolution, it tells you there had to have been fresh water coming into the system, because brine that's been in contact with salt usually is saturated. So you had to have reintroduction of fresh water. This is a little different of a marker. There's not much bromide data anywhere. So we use sodium to chloride ratios, and halite has a ratio of 1. So if you see a brine that has a ratio of about 1, that tells you halite dissolution. And I just want to point out that we see it in the bottom formations, the Mississippian and the Devonian, beneath the salt at the bottom of the Paradox Basin, and it's pretty much everywhere. So it tells us massive influx of relatively dilute groundwater to depth. You can actually see this salt dissolution at the surface today. So does anybody raise your hand if you've ever been to the Dolores River or the Paradox Valley? Okay, yay, it's one of my favorite places. It's important for society, and I'll tell you why in a minute. But you can see hydrology in action. You can see the snow-capped peaks of the La Salle Mountains. That is the water tower where most of the recharge happens. Flows underground through the sandstone aquifers, and then it comes in contact with that 3-kilometer salt wall underlying the Paradox Valley. It dissolves the salt at pretty shallow depth, and it discharges into the Dolores River. You can see the salt crust along the edge of the river. The reason we should all care, and we've dated it, and it takes about several thousand to tens of thousands of years for that water to circulate. Well, this is a major source of salt to the Colorado River. The Dolores River is an important tributary to the Colorado. The United States has a treaty with Mexico that we have to deliver a certain amount of water, but also of a certain high enough quality. Well, this is one of the main sources of salinity to the Colorado River. It's called geologic sources of salinity. In order to mitigate or lower the salinity on the Dolores River, the Bureau of Reclamation has these brine pumping wells. It's a shallow brine body. They have 9 wells that they pump out that brine pretty continuously. They were re-injecting it beneath the salt to the bottom of the basin in the Leadville limestone. It was causing induced seismicity, earthquakes. So I've actually, last I heard when we were up there last summer, they had to stop injection. So they're storing the brine at the surface. But it also has an interesting biology story because that salt is not just halite, it's also gypsum. And gypsum is important because when you dissolve it, you provide sulfate, which is an electron acceptor, and this salt is interbedded with organic rich shale, which is the electron donor. So microbes love to use that and reduce the sulfate and produce hydrogen sulfide. There is over 200 ppm of hydrogen sulfide that is biological in origin out of these wells. And so we've sampled the biology. We know from the sulfur isotopes, it's all biological. So we know something about the time scale of circulation of groundwater and salt dissolution, and because we date the water. So I'm going to start with the top. You will not see depth plots for the Paradox Basin in anything I do because the structure is so complex that with those salt diapirs. So at the surface, it's essentially in stratigraphic order to the bottom of the basin. So this is a picture of my former PhD student, Ji-Hoon Kim, who did a lot of this dating work and on the brine chemistry. Ji-Hoon went on to a postdoc at University of Calgary and is now a research scientist in Korea at the Korea Institute for Geology and Mineral Resources. So I'll start with the shallowest. Again, those salt anticline brines that they're pumping out, again, are on the order of thousands to tens of thousands of years. And that's based on carbon-14 dating, as well as tritium helium that we send our samples to Kip's lab to analyze. The fresh to brackish groundwater in the Cretaceous units and the Navajo sandstone are also on that time scale of thousands to tens of thousands of years. And you do see Pleistocene groundwater retained in that. But if you go a little bit deeper into the fluids produced with the oil and gas, I was expecting to see groundwater age increase with depth. That's kind of the paradigm in hydrology. And I was shocked to find less than 1 million year old water down 3 kilometers beneath the salt. And we determined that based on a relatively new age tracer called Krypton 81. Comes from the atmosphere, there's no subsurface sources, and it radioactively decays. So at first I thought, oh wow, we did something wrong, there's no way. But I'm gonna show you a second line of evidence that does support this idea that there is relatively young water at the very bottom of the Paradox Basin. Within the Paradox Formation salt, in that shale interbed, it was beyond the dating limits, which I would expect. I'll show you more evidence that the salt's relatively impermeable. And the way that we date really old water is to use helium-4. Again, this is something that Kip is more of an expert on than I am, but helium-4 ages were much older. And that's because helium is coming from the underlying basement rocks and accumulating in this basal aquifer. So there's a helium resources story that I'll return to at the end of the talk. So how do we get less than 1 million year old water to the bottom of the Paradox Basin? I'll come back to, and I'll give you a hint, has to do with the Colorado River incision. But as a second line of evidence that this has really happened, we turn to what I call fancy noble gases, where you essentially look at every isotope ratio of every noble gas. And this was done by Rebecca Tyne, who is a PhD student of Chris Ballantyne, and now she's an assistant professor at the University of Manchester. Interestingly, what she found is in the basal aquifer, so the Leadville limestone and the McCracken, and some of the formations above the salt, it looks like the helium that accumulated over geologic time and the water that was equilibrated with that has been flushed out by new meteoric water that's come in the last several million years and is now re-equilibrating with that helium that's remained as a helium cap. So we now have noble gas evidence of massive flushing of that basal aquifer in the last few million years. We also, interestingly, in those shallow salt diapir brines in the Paradox Valley, there's been so much hydrogen sulfide produced by microbes that you actually have helium exsolving into that gas cap of H2S, which I thought was really interesting. In terms of resources, there's a lot of locations in the Colorado Plateau where helium has accumulated. This would not be a good place for helium accumulation because if you have massive flushing by meteoric recharge, you would have flushed out a lot of the helium that had accumulated over geologic time. So we've been kind of using these approaches to try to look at prospecting for helium accumulation. So how do you get fresh meteoric water down 3 kilometers in the last few million years when the basal aquifer is actually pretty thin? You have topographic gradient with the La Salle Mountains and other laccoliths. But really the short of the story is that you have to have relatively high permeable basement rocks underneath these basal aquifers. My colleague Mark Persson is the one who makes these types of hydrologic models. And what he did is he tried to simulate what we saw from the brine chemistry. And the only way that he could get meteoric water to the bottom of the paradox basin, flushing those brines and helium, was to enhance the permeability of the basement rock. Which is interesting because as hydrologists or geologists, we often make basement rocks impermeable. And we just say nothing happens beneath that. But those basement rocks were exposed at the surface at one point in the geologic past, and they were a critical zone, and they did weather. So they do tend to have enhanced permeability. So from a hydrologic modeling perspective, that helped to get recharge from the mountains down. And I should also mention that the mountains, they're not only the water tower, but they also go into the subsurface. And so you can have mountain block recharge down 3 kilometers, and that is the source of the water. So the modeling predicts that we can have deep circulation through the mountain blocks, but how can we tell from the chemistry in the water? So there was this really interesting paradigm kind of set up by Laura Crossie, who I'm a big fan of, at University of New Mexico. In the Grand Canyon area, she has sampled all of these springs that are emanating from the base that are really radiogenic. So they have a strontium isotope signature that doesn't reflect the sediments. It has to be coming from really ancient rocks beneath that, the Colorado Plateau sediment. And so she suggests that recharge in the San Francisco peaks goes down deep faults, interacts with the radiogenic rocks, and then emanates in the springs. We think we see the same thing in the Paradox Basin. If you look at the strontium isotope signature of brines that are in the formations beneath the salt up against those basement rocks, they're far more radiogenic than the sediments. So it provides some evidence that those fluids have interacted with the underlying basement rocks. So I want to get to the microbiology story. So we had this hypothesis in our SMRFs proposal that all of that deep meteoric circulation that has flushed brines may have brought microbes from the surface down into sediment that had been previously sterilized. And that if that deep meteoric circulation was driven by incision of the Colorado River and denudation, this is a pretty recent phenomenon. And so thus maybe led to a recent bloom of microbial activity in the subsurface. So to do this work, again, through the CFAR project, I've now been working with Maggie Osborne, geomicrobiologist. And does anybody recognize where this spring is or what that is? I've been told today that several of you have been there. What's that? Onion Creek, yeah. So this is a sulfur spring emanating from the salt diapir from gypsum dissolution. Microbes making H2S for bacterial sulfate reduction. Yeah, so Maggie and her PhD student Mira Shaw were collecting microbes from the water that we dated and did chemistry. From the oil and gas wells, it's pretty dirty work. So we collect the brine that comes out with the oil and gas. It's mixed, you let it sit in a carboy. If you're lucky, the oil floats to the top and you collect the brine from the bottom. And then we used to do it by hand with a bike pump. Here's Jackson Watkins, a PhD student who's actually since left to become a real rock star. So in Chicago, he's in a band now. But he is passing that brine through filters, and that's what we're measuring is the DNA and RNA, as well as culturing. And now we have an automated system. Yeah. So this is trying to show you, this is elevation, but you could think of it as stratigraphy as you go deeper and deeper. And this is the number of DNA reads that Maggie was able to sequence from all of these fresh to brackish aquifers all the way down through the oil and gas wells. And I'm just going to highlight the fact that everywhere we looked in the Colorado Plateau, we found microorganisms. So even in sediment that had reached really high temperatures, so the paradox or Hermosa Formation salt, although not many, that's the Aneth oil field. Lots in the Leadville, limestone and McCracken at the bottom of the basin, and then lots in the Honaker Trail and shallower sediments. And I just wanted to shout out to some of the industry partners because this work would not be possible without sampling their wells. So we worked early on with Paradox Resources. It's no longer in business. ARB Energy, Running Foxes, and the Navajo Nation Oil and Gas. Okay. So we find microbes everywhere, including in sediment that was likely sterilized. We can also somewhat tell what these microbes are doing by the things that they produce that are left behind in the water. And so, for example, we can see formation water produced with oil and gas in the Dakota, Morrison, and Mesa Verde formation, pretty shallow. And this is actually up into the Uinta Basin. Has really high dissolved inorganic carbon and also really enriched in 13C. This is an indicator of microbial methanogenesis, which is one of the most common microbial processes in the subsurface. Maggie has taken all of that DNA, RNA, and has been able to show for individual wells, which I won't go into, that there's lots of microbes that are doing important, interesting things like degrading oil in black. They're reducing sulfate to make H2S, which we see, and lots of microbes that are methanogens that are making methane through several different pathways. So. We can see that microbes are cycling carbon and sulfur, mostly in the relatively shallow reservoirs today, from the microbiology. And what we're trying to do right now is pair that to the isotopic signatures. So I just want to show you evidence of microbial activity from the things they produce, which is more my wheelhouse. So this is showing clumped isotopes of methane versus the hydrogen isotope of methane in the water. I'm not going to get down into the weeds, but I just want to show you, based on clumped isotopes, we can tell that there are microbes within the Cutler Formation, the Honaker Trail, and the Leadville that are chomping on methane. They are microbes that anaerobically oxidize methane. And we see that because they have this really anomalous signature in clumped isotopes that plots above the equilibrium line. So microbes are oxidizing methane. They're making methane in other places. And they're also oxidizing higher chain hydrocarbons. So there's lots of thermogenic gas because when that oil formed, you also formed gas, which contained methane, ethane, propane. And we can see that after that gas and oil migrated out of the source rocks and into shallower reservoirs like the Honaker Trail and Cutler, we can see that there are microbes that have oxidized that, in this case, ethane, because we see that the concentration of ethane, it's been consumed for the most part, and we can see that the residual reservoir is really enriched in the heavy isotopes because microbes like to break the bonds or eat light isotopes. So lots of evidence of microbes doing, making methane, oxidizing methane, making hydrogen sulfide, but we would argue in the relatively recent geological past. So I'm going to summarize this first part of the talk as kind of the big picture basin scale, where at the maximum burial, things were too hot for microbes, but you generated hydrocarbons, and they migrated into shallower reservoirs, which set the stage for later microbial activity. So it could have created a shallower hotspot for microbes that cycle carbon. What we see today, when the Colorado River started incising and denuding the landscape, a lot of those oil and gas reservoirs and the residual bitumen is left closer and closer to the surface in contact with circulating meteoric water, which likely reintroduced microbes and led to things like bacterial sulfate reduction, methanogenesis, and anaerobic oxidation of hydrocarbons. And then I end with this little section with what we see today in places like Rainbow Rock, where some of those hydrocarbons are actually above the water table. And I call that the last gasp. You know, they could have been oxidized in the recent past. So since that study, Mira Shah, who's a PhD student of Maggie's, has done some really great, very detailed organic geochemistry work that I'm not going to get into the weeds because even I don't understand it. But by looking at the ratio of organic compounds, she's able to better tell the extent of biodegradation. And in places like Rainbow Rocks, where you would expect a lot of biodegradation because it's even exposed at the surface today, or the tar sands and the white rim sandstone. And what was really shocking to us is that they are biodegraded, but not to the extent that we would expect, and not as much as some other oil and gas reservoirs around the world. And so our working hypothesis right now is that maybe the last few million years of denudation has not been long enough. That it's just started this process of microbes being active in the subsurface, and that it's going to take another few million years to be able to degrade that oil. So everything I've presented so far is what I'd call more basin scale. In this SMERS project, Henrik Drake really drove a lot of this work. We were able to find what I call a needle in the haystack. We went looking for calcites that are precipitated along fractures that could maybe tell us the history of past fluid rock reaction and microbial activity. This is one of our favorite samples. You can see it's coarse grain calcite. If you zoom in, this is a single grain of calcite. Henrik, using a technique called nanosims, was able to measure the isotope composition of individual calcite growth bands or precipitation events. And using uranium lead dating, we were able to get over 100 million years of history in this one calcite grain, which blows my mind. In the first 100 million years, up until about 5 million years ago, there's no evidence of biology. And the oxygen isotopes of the calcite look like basinal brine, evaporated seawater. Clumped isotopes are relatively warm. And then something happened, again, about 5 million years ago, which coincides with incision of the Colorado River. All of a sudden, there's this massive enrichment in C13 of the calcite, and that screams of microbial methanogenesis. So it looks like about 5 million years ago, things turned on in terms of biology in the subsurface. And we again think it's because of circulation of meteoric water. So we were all excited about that one sample, but can we replicate it? And so we've been measuring additional calcites, and we found a few more. We've also been measuring barite, because just like I showed you, microbes cycle carbon, they also cycle sulfur. So by measuring the sulfur isotopes of the barite, we see that there are 2 generations of barite. And interestingly, this blue generation has some of the most negative sulfur isotope values that have been published. And I'll show you what Henrik thinks, why they have that value. And the range of temperatures go from more recently down to at least 60 degrees C, which is at least more hospitable. So this is Henrik's kind of hypothesis of what maybe happened in terms of the barite, is that early on, you had barite forming that has a sulfur isotope signature that looks like seawater. Would have been that evaporated seawater brine. And then during the Miocene and Pliocene, maybe there started to be some denudation and you started to bring that sediment closer to the surface, you could have sloshed some near surface water that could have brought microbes into the subsurface, but still anoxic. So pyrite could have formed by biology with a really negative sulfur isotope value. And then that pyrite, most recently, was oxidized to form the barite, which still carries that really depleted sulfur isotope value. So now we've got some calcites, and now we've got some barites that we're pretty excited about, that we can tell the basin history. So Henrik has this much fancier block diagram of the history of the Colorado Plateau, which I'm not going to go through in detail, but just to say that you had early sedimentation and maximum burial, which we can now see in these calcite grains and barite that show that this was a time when the basin was hot, it was saline, and it was likely abiotic. Starting at about 13 to 10 million years ago, there started to be incision of the Colorado River and denudation, which unroofed the basin, brought sediment to closer to the surface, cooler temperature, circulation of meteoric water, and all of a sudden we start to see biological signatures in the calcites and barites, as well as in the fluids remaining in the basin. So I'm going to end my talk with, try to go through this quickly, of what does this mean for resources and the climate crisis? Well, the residual evaporated seawater brine is enriched in lithium. It's not as high in the Paradox Basin as it is in places like the Smackover Formation in the Gulf of Mexico, but it is over 100 ppm in some locations. And if you look at lithium concentrations in brines, it's highest in the formations that still contain the evaporated paleo seawater. So the Paradox, the Leadville, and the McCracken Formation. And that's what's being targeted for the exploratory lithium mining. Lithium doesn't just come from the evaporated seawater, the residual brine. Lithium is far more enriched than evaporated seawater. And so we know there has to be an additional source, likely from fluid rock reaction over geologic time. And we can tell that in part using our favorite tracer, chloride to bromide, that lithium is highest in the Paradox Basin associated with that evaporated paleo seawater. But where did it pick up that extra lithium? Likely from the shales within the salt. So we've measured lithium concentration in the shale. It's elevated. It's likely through desorption added lithium. This was work done by my former PhD student, Mohammed Marza, who's now an assistant professor in Kuwait. I'm not going to go through this, but Ji-Hoon Kim, with her research scientist position, is doing all sorts of interesting work now looking at lithium sources and lithium isotopes through rocks that we collected at the Utah Core Repository. So you're really lucky that you've got rocks that are stored just downtown in Salt Lake City, that we were able to go and actually collect the shale from the Paradox Formation and be able to do sequential extractions to see how much lithium is there, where did it come from, how might you release it for production. And finally, we've been working on helium resources. So I mentioned the areas that we've been working are outside of what are the traditional helium accumulations. So they're places that are a little more open hydrologically, and helium that's accumulated has been flushed in a lot of places. And we can see that because we see helium coming from the basement rocks, accumulating beneath the salt, but again, some of it's flushed. And interestingly, there's very little helium diffusion through the salt. So that tells us that that salt is a great seal for other places where helium's been retained. And that's where you would explore for it. And so right now we're working up in the Cisco field with a company called Running Foxes on helium accumulations. So my very, very last slide, I promise, is I want to end on education and public outreach because it's something that I think is really important and I'm passionate about, starting with our critical zone observatory. So I don't know if you're involved, Paul, but they set up something called the Earth Science Discovery Program. It's mostly for 3rd through 6th graders, mostly Title I schools, so underserved in Tucson. And they bring about 300 to 600 school kids to our science center on campus. And they have all these different hands-on stations. So they learn about the rock cycle, they learn about the water cycle. So we added a station from our SMERS project on subsurface microbiology. So I drew this crude cross section, and we took all those microbes that Maggie Osborne discovered, and we have the students adopt a microbe and they learn about their habitat. And I'm so proud because my daughter, Emily, who tends to be pretty shy, she got up and presented her microbe and she chose a methanogen. So, and we have this cool thing where they put their microbe up here and actually shows diversity decreasing with depth with their habitat. And then my son, Bodie, had done the program. He's now a 6th grader, a middle schooler, and Emily is a 5th grader. And so his class came. And the students that are the facilitators are amazing. They're students across campus, so not only geosciences, and they provide awesome mentors to these school kids and show them that scientists aren't your traditional model in a lab coat in the lab. So thank you for your attention, and I hope there's a few minutes for questions. Yes, Kip.
Great. How do you get these microbes down to, we use sand as filters and to transport abundances that you need via the primary permeability, or refractions? How do you get them down?
Yeah, that's a really good question. And I just submitted a paper on permeability and microbes. So in a sandstone, there's typically enough permeability porosity. It really comes down to the pore throat size. So you can have big holes, but if they're restricted to move. So it looks like in the upper about 1 kilometer of crust, you tend to have enough permeability in sandstones and limestones. And a shale, it tends to be the fractures. The matrix is too small. And there's a lot of studies that show that. And then in general, as you go down with depth, because of diagenesis, compaction, you tend to decrease that pore throat size. So we think permeability is actually a major control on why you get less and less biomass as you go deeper and deeper. Yep.
All the inactive microbial, but I find it tied between the activation of
yeah.
I think
yeah, I think you're absolutely right. And you sound like you're more of a microbiologist than I am. And that's why I think I have to be cautious, because they really could have been deposited with the sediment and then been somewhat dormant because they are resilient. And another thing that maybe you're alluding to is it doesn't have to be transport from the surface. There is, water that would have been kind of intermediate that could have been brought in. So they don't have to be transported all the way from the surface, but it could have come a little bit up gradient, maybe never reached those high enough temperatures. And I think that's a really challenging thing. I'd be curious if you have ideas how you could tell that. Yeah. Okay, cool. Yeah.
So with the well data and the microbiome, how concerned we need to be about
Yeah, that's a great question that plagues any subsurface microbial study. And basically what we do is, we, meaning them, is you can't get away from contamination. It's always there with drilling fluids. So the best you can do is sample the drilling fluids, sample the subsurface, and then look for similarities or differences. We did in that study sample separators and tanks, compared to wells, because in many places you actually can't get wellhead samples. And we show that there's a lot of similarities we'd expect, but there's blooms in the tank because it's warm and they're happy. And yeah, but that's an issue we struggle with, but we think that we can discern microbes that have been in the subsurface for a long time. Yeah. Eugene.
Yeah, I've been thinking a lot about mapping geothermal resources across the state. So I'm going to ask a question. But whether or not the microbes have been reactivated or flown through, the fact that they are active in that part of the subsurface, is there anything you can say about temperatures at those stratigraphic horizons modern day? So activity.
Yeah, that's interesting. It seems like a more challenging way than just measuring the temperature, but
for sure, all your samples for some of
yeah.
Outflow and springs. That would be a case where there would be a spring.
I see.
Outflow measurement.
Yeah, that's an interesting idea because maybe you have low temperature at the surface, but the microbes had experienced something hotter. Yeah. Yeah, I've never thought about that in a prospecting way. I was telling somebody else that my favorite studies were done by actually Michelle Walvrod with the volcanic intrusion. So there's a lot of studies of where hydrothermal activity has sterilized the surrounding rock and then microbes have been brought back in. But I'm not familiar with what you're talking about. Yeah.
Lithium. Mm-hmm. I like the shale. I know something about the base. And then have all
That's interesting. And now I remember you're the lithium guy. We did the National Academy of Sciences talk together. So the basement is really interesting. I think what you're alluding to is that in this Smackover Formation, which is the basal aquifer in the Gulf of Mexico, there's a lot of evidence that geothermal fluids brought lithium because high temperature reacting with pegmatites and things brought lithium into those brines. There's elevated temperature, there's thermochemical sulfate reduction. We don't see that in the Paradox Basin. And it does look like the highest lithium is within the Paradox Formation salts, the shales interbedded. I did think about that and look for that. And you also see the hydrothermal over in the Clayton Valley, and you see it in South America. So it does seem like it's important for the biggest lithium resources. Yeah. And I don't know why we don't have that geothermal. And I'll look to you, Eugene. There's a reason we don't have it here. Yeah.
Colorado Plateau.
Mm-hmm. Yeah. Yeah. You are far more of an expert on structure than I am. But what I wanted to show you is that, yeah, at first I thought this seemed implausible, but because of that structure, I don't know about the timing. Okay. You'd have to ask Bob Krantz and other people on our project. But if you look at the basement rocks are actually just juxtaposed against the sedimentary units. So I don't think you have to go too far to have interaction. And I don't think it's fluid trapped in the basement. I think it's basinal brines that have circulated. Because you see that also in the La Salles in the hydrothermal system. It's basinal brine that's circulated. It's not like volcanic fluid that's gone out into the sediment. These are awesome questions. Thank you.
Thank you for next.